Hi biologists. So what we're going to look at today is how an electrical impulse travels down a nervous cell, okay, so a neuron. So last lesson we talked about how sensors detected changes in the environment, but now we're going to look at what actually happens to pass that electrical impulse along the axon of the neuron. So if you look at the first slide, what we've got here is we've got the plasma membrane. So the orangey bit in the middle is the plasma membrane, that's the phospholipid bilayer. And you can see there that we've got three sets of protein channels within the membrane. We've got a potassium channel, we've got a sodium channel, which is often referred to as a sodium ion gated channel. It's important that you know the word gated. And in the centre, the red one, we have a sodium potassium pump, and because that is a pump, that requires the use of ATP. The potassium channel and the sodium channel do not require ATP at all. Okay, and so what happens is for the first part of this, we haven't got an impulse at the moment, we're just at rest, so the neuron is just there, it's not having an electrical impulse passed along it. But the sodium potassium pumps are working, so they're using ATP all the time, and what they are doing is they are actively transporting sodium ions, so Na+, out of the cytoplasm at the bottom of the picture, and they are actively pumping potassium ions into the cytoplasm. Okay, and for every 2 potassium ions that they move 3 sodium ions. So if we look at what's happening with the sodium potassium pump, we're pumping 3 sodium ions out of the cytoplasm, out through the plasma membrane, and we're pumping 2 potassium ions into the cytoplasm. And what that does is it creates a gradient of sodium ions across the membrane. So we have a lot of sodium ions on the outside and we don't have very many on the inside in the cytoplasm. Because these are ions, we call this an electrochemical gradient. So we've got a sodium ion electrochemical gradient, but we also have a potassium ion electrochemical gradient because we have lots of potassium ions on the inside in the cytoplasm and we don't have very many on the outside, out on the other side of the plasma membrane. So because of that electrochemical gradient, the ions want to diffuse back to the opposite side. Now they can't get through the bilipid layer. The only way they can get through the plasma membrane is through either the potassium channel or through the sodium channel. Now the sodium channel is also referred to as a sodium ion gated channel, and it's important that you know the word gated there. Now the problem is that they could, if those channels were open, then the ions just could diffuse through to restore the gradient and get it back to equal again. But the potassium ion gradient or channel, sorry, is open. So those potassium ions are able to start to move back to the outside, but the sodium gated channel is actually shut. And what that means is that all of those potassium ions now also start to flow through that potassium channel to the outside of the cell. So we've got a high concentration now of sodium ions, and we've got potassium ions on the outside. On the inside, we just have some potassium ions. So that means that the outside is now really quite positively charged compared to the inside. The inside still is positively charged because we've got potassium ions, which are positive, but because we have many more positive ions on the outside, because we've got all those sodiums and we've got extra potassiums now, it means that the outside of the membrane is relatively more positively charged than the inside. And that gives us a potential difference across the membrane. A potential difference is basically a voltage where we would have a difference in charge across the membrane, and the resting potential is actually calculated to be minus 70 millivolts. So that is the fact that we have relatively more positivity on the outside of the membrane than we do on the inside. Because we have this potential difference of minus 70 millivolts across the plasma membrane, we say that the membrane is polarized, okay, because it's relatively positive on the outside compared to the inside. Okay, so we have established our resting potential of minus 70 millivolts, and that is the normal state of axons. But then you have a stimulus, and what the stimulus does, if you like touch something or something's hot or there's light reflecting on your retina, for instance, then what will happen is that will cause some of those sodium ion gated voltage channels to open. And so if you look on the diagram here, the green channel has opened, and because we've got this electrochemical gradient across the bilipid layer, across the plasma membrane, it means that sodium ions will diffuse through the gated channel to the inside of the cell, into the cytoplasm. And as they do that, they're bringing obviously positive charges into the cytoplasm, and that means that now what happens is it triggers other gated sodium voltage channels to open. So we have positive feedback there. So positive feedback is when we have one event which triggers the event to be enhanced further. Okay, so one channel opening triggers many more to open, and so more sodium ions pass into the cytoplasm. So that causes like loads and loads and loads of sodium ions to flood into the cytoplasm, and because we've now got all these positive charges passing into the cytoplasm now becomes relatively more positive on the inside compared to the outside. So the charges have switched across the membrane, and at that point we say that the axon is now depolarized, and we call that an action potential. So once we get a voltage of plus 40 millivolts across the bilipid layer, across the membrane, then those sodium ion voltage gated channels close, and the potassium ion voltage gated channels open, and that allows those potassium ions to diffuse out of the cell. And so what we're then doing is we're losing those positive ions now from in the cytoplasm. They're flowing back to the outside, and so now we get the situation where we have a reversal again of the charge across the membrane. So now we have it becoming more positive on the outside compared to the inside. Because we have that sudden flood of potassium ions out across the membrane from the cytoplasm to the outside of the cell, there's a massive flood, and it overshoots back to its minus 70 millivolt position, so it goes beyond that, and we call that hyperpolarization because it's more negative than we would usually have in the resting state. So once we've reached that point there, the potassium ion gates now close, and the sodium potassium pump now returns to its pumping 3 sodium ions out, 2 potassium ions in using ATP. And that is referred to as repolarization, because what we're doing now is returning our membrane to that minus 70 millivolt position back to its resting state.